The Nobel Prize Session, sponsored by the World Gastroenterology Organization Porto Training Center, is about to begin. The Nobel Laureates are Professor Harvey Alter and Professor Michael Houghton. The chairpeople are Professor Guilherme Macedo, President of the Portuguese Society of Gastroenterology, and Professor Fátima Carneiro, Director of Anatomical Pathology at the University Hospital São João.
Welcome to you all in this Nobel Prize session, which is an extremely honorable event for us. I have to tell you that perhaps this is the most striking and most important event that we have been in the last years, and I can assure you that we've been in many this type of events, academic events. Now, first things first. First, to congratulate the organizing committee of this 16th YES meeting of this brilliant event, as it is right now, and also for mounting up this session, which, as you will see, it will be extremely interesting, important, and relevant to us all. The second word, of course, will be for Professor Fátima Carneiro. She will share with me this session. So today you will have one great pathologist and one modest hepatologist, which is me. But anyway, this is what we can get. But anyway, the most important thing today and what we are going to hear will be two magnificent presentations of our invited today, because today, you will hear the amazing story of hepatitis C. And throughout the next minutes, next hour or so, you will hear the amazing journey of these two very important people in our world that really changed the landscape of how we see not only this virus, but in general, liver diseases, for example. So, It is my pleasure, of course, and my honor to present Professor Harvey Alter. He, as you know, he's been nominated and won the Nobel Prize last year when we were talking about the other pandemic, which is the COVID-19. But for long, we had for decades the other pandemic, which was hepatitis C. But in fact, Professor Harvey Alter has been designated a Distinguished NIH Investigator. There are only 23, by the way, 23 scientists that hold this distinction. He has a very long career in clinical research and played a very important role in the discovery of two hepatitis viruses, namely the hepatitis B and the non-A, non-B, later designated the hepatitis C virus. And I was there when he presented the his first works in US. I was a young student, well, almost student. I was a young resident anyway, but I was there, and I remember the thrills and the emotions that we had at that time. He managed to conduct many long-term perspective studies, and he helped to define the natural history of the non-A, non-B infection, which was then shown to be the hepatitis C infection. So he was the principal investigator in many prospective studies, especially those related with transfusion associated, where that became a very important hallmark of the story. And in fact, that has made a great changing in the way as we see and how we treat our patients nowadays. For these studies, Professor Alter has been awarded the PHS Distinguished Service Medal, the first international medal for sciences from Francis Anselme, the American College of Physicians Award for Outstanding Work in Science, and of course, the Distinguished Achievement Award of the American Association for Study of Liver Disease. He also has been elected to fellowship in the American Association of Physicians and received several other prestigious awards. And finally, as you know, he was awarded the Nobel Prize in Physiology and Medicine in 2020. So, Professor Harvey Alter, welcome virtually, and we are eager to hear from you your presentation. And thank you.
Really is very indicative of what I'm going to say, because I am going to show you thank you. The title of my slide, my title of my talk really is very indicative of what I'm going to say, because I am going to show you how we got to the end of the beginning of hepatitis C and how we might get to the beginning of the end. This is the way we are right now. This is the way we are in this time of COVID. And I'm so sad that I'm not in Porto right now. COVID has prevented me from being in many beautiful places, and Porto was one I was really looking forward to. But I'll do my best to do this virtually. When I received the Nobel Prize, I received this card from a friend that I thought was very profound. And it said, "There is no elevator to success. You have to take the stairs." And I think if there's any message that I leave with you today, this is it. And I'm going to show you the stairs we took towards the discovery of hepatitis C. And it began actually long ago. The history of hepatitis goes back to 400 BC, the time of Hippocrates. Hippocrates was a philosopher, a physician, and a great observer. And he observed that people were getting yellow at times, which he called icterus, and also that they developed hardening of the liver that he called cirrhosis, which we now call cirrhosis. The Hippocrates thought that this was due to an imbalance of the four humors: black bile, yellow bile, phlegm, and blood. And while this didn't hold up to peer review over the years, he made a profound statement, and namely that disease was not due to divine punishment, as was believed at the time, but rather it was due to some external force that was perturbing these four humors, in this case, an excess of yellow bile. Nothing much happened over the next 2,000 years from Hippocrates, except that there were recurrent wars. Sorry. There were recurrent wars, and in every war, many combatants came down with jaundice. This is called campaign jaundice, and then sometimes vaccine jaundice when it was shown due to vaccine, and ultimately it was distinguished as hepatitis A, infectious hepatitis, or hepatitis B, serum hepatitis. But throughout those 2,000 years, there was no indication of what the agent was, or how to test for it. The breakthrough came in the early 1960s when, fortuitously, I was working at NIH with Dr. Brooke Bloomberg, and while we were looking for lipoprotein polymorphisms in human blood, we discovered a line that did not take up the lipid stain of the lipoproteins, but rather stained red just for protein. This turned out to be a reaction in agar between the serum of a multiply transfused patient with hemophilia and an Australian Aborigine who just by chance was being tested on that day. This came to be known as the Australia antigen, and ultimately Bloomberg traced this down and linked it to hepatitis, and it ultimately became known as the hepatitis B surface antigen. But this was the first true marker for any hepatitis agent, and also was the basis for the first hepatitis B vaccine. The story I'm really going to tell begins in around late 1960s, when at NIH, Paul Holland and Paul Schmidt and Bob Purcell started the prospective studies of patients who have open heart surgery. In 1970, I came back to NIH and took over these studies. The key was that we used open heart surgery patients because they received a lot of blood. The heart-lung machine at that time required 7 units of blood just to prime the pumps. But importantly, we sampled patients before transfusion and then every 1 to 2 weeks post-transfusion. We saved these samples in frozen storage and went back to them many times as better assays became available. But initially, we only had one assay that could detect hepatitis, and that was the serum enzyme alanine aminotransferase, or ALT. And shortly thereafter, we were able to measure hepatitis B surface antigen. We also saved the donor samples and could link those for later testing. What was dramatic was that at first we found that prior to 1970, that about a third of our patients were developing post-transfusion hepatitis. And this was quite startling. And John Walsh at that time did a study and showed that the main reason for this was that about half our patients were receiving blood from paid donor sources. And that if you received at least 1 unit of blood from a paid donor, there was a 51% chance of developing hepatitis. Whereas if you received only all volunteer donor blood, there was only a 7% chance. Thus, in 1970, we switched to an all volunteer donor system and introduced the first and then second generation test for hepatitis B surface antigen. This caused a precipitous fall in post-transfusion hepatitis, bringing the rates down to about 10%, even though the number of units transfused remained almost identical. The next step was that in 1973, Abbott Laboratories developed a sensitive third generation test for hepatitis B antigen. We went back and tested these stored frozen samples and was somewhat surprised to find that hepatitis B represented only about 25% of the total hepatitis, and that there was some other non-B entity which was the major cause of post-transfusion hepatitis. Then in 1975, Feinstein, Kapickian, and Purcell, this is Steve Feinstein, Bob Purcell, and Al Kapickian, discovered the hepatitis A virus using immune electron microscopy. Since these were our collaborators here on the NIH campus, we immediately tested these non-B samples for hepatitis A and found that not a single one was due to hepatitis A virus. And it was then in a brilliant bit of deductive reasoning that we said that these were not hepatitis A and not hepatitis B, we'd call them non-A, non-B hepatitis. We didn't like this awkward nomenclature, but we hadn't yet proven that we had a virus. And if it was a virus, we didn't know how many there might be. So we held off calling it hepatitis C. And this non-A, non-B entity lasted much longer than we thought. The next step was the chimpanzee model. This was an invaluable model. Many think that chimp should have been the fourth winner of this Nobel Prize. Chimps did not get harmed in these studies. In fact, they loved being in our studies, and you can see them here lining up to sign their informed consent. But with the chimp, we were able to learn a lot. And we were able first to show that we could transmit this non-A, non-B hepatitis from patients who had acute non-A, non-B, and from patients who had chronic non-A, non-B hepatitis, and importantly, from totally asymptomatic blood donors who had been implicated in hepatitis transmission. The next important step, well, first, I just want to make this point, because I work for the government, people who work for the government are thought to be lazy and to just live off the government dole. But I just wanted you to know that I always gave 100% at work, still do. It's just that it has this peculiar distribution. Today is Monday here in the US, so it's not one of my days. Next came this important patient, Mr. H. Mr. H was a man who was very vigorous, who loved to climb mountains and blaze trails. And one day, while up on one of his treks, he had a cardiac arrest. He would have died there on the mountain, except for the fact that his wife was with him, and she administered CPR and was able to revive him. He eventually made his way back to NIH, and this shows his clinical course. Just focus now on this blue area. These are his ALT elevations. You see he was transfused here at 0.0, that at week 5, his ALT levels began to rise and then rose very rapidly up to a peak of 2,112, where the upper limit of normal was just 40. At that time, he was jaundiced and quite sick. However, he recovered rapidly. Enzymes came down fast, and the story went on that he recovered and became a chronic persistent carrier for the rest of his life. But we were able to obtain a plasmapheresis unit at point A. And that was put into a chimpanzee and was infectious in the chimp and was titrated out by Bob Purcell to have a titer of 1 × 10⁶.5 chimp infectious doses. We'll get back to that later. At point B, we obtained another apheresis unit, but this was put into a second chimp and it was not infectious. The reason we think it was non-infectious because when we later went back and measured Hcv RNA, as shown by this yellow line, you can see the viral load was falling rapidly, and also that antibody had appeared by that point, and the virus was then immune complexed and thus less infectious. This is shown on the bottom panel, but we don't need to go into that. What's interesting, if you follow this Hcv RNA curve, is that right after transfusion, there was a little blip. This was the virus coming in from the donor that then was somehow contained immunologically, but then probably through a breakthrough infection, rose again to the peak level of 3 times 10 to the 7th copies per ml, almost the same as the chimp infectious doses. This material was then titered in chimps, and because we now had a titered infectious inoculant with a known infectious titer, we now had two things. We had this material from Mr. H, and we had the chimp model. Thus, Steve Feinstone did this experiment because when you had these two pieces, you could manipulate the system. And he did a chloroform extraction of this putative non-A, non-B agent and showed that it was no longer infectious after chloroform treatment. Whereas if he did a sham extraction, it caused hepatitis in the chimp. Thus, we knew at that point that the virus had to have essential lipid in its envelope because chloroform is a lipid solvent. So we now knew it was an envelope agent with lipid in the membrane. Then went on to do studies with filtration, and I won't get into those, but you could show that the virus was quite small. And with these two things, we're able to say that this was a small agent, somewhere around 30 to 60 nanometers in diameter, that it had a surface coating with essential lipid. And this narrowed it down to these candidate viruses. Either it was going to be these small RNA viruses, so-called alpha or flaviviruses, or it's going to be a hepatitis B-like virus. But we had a lot of evidence that it was not related to hepatitis B. And thus it got down to being either a new class of viruses or one of these small RNA agents. It was Dan Bradley at CDC who said the data were most consistent with this being a flavivirus, as it eventually proved to be. Now, over the years, while we tried to develop a specific test for this agent, which we were unable to do, we went on to do clinical studies by doing liver biopsies with the NIH liver unit. And we initially biopsied 39 patients and showed that while most had pretty mild disease, 10% already had cirrhosis, and another 13% had what was called severe chronic active hepatitis that would probably progress to cirrhosis. We then did follow-up biopsies in 20 of these patients, and while most improved or were stable, 25% progressed to cirrhosis under observation, and thus we had 8 out of 39 patients, or 20%, that developed cirrhosis in the study. Now that figure of 20% has pretty well held up over the years. The rate of cirrhosis is somewhere between 20 and 30%. Importantly, we showed that 3 of these patients died of liver failure, and 3 others had very severe liver disease when they died of the underlying heart disease. So we now knew that non-A, non-B was not a benign disease, as some thought, but rather it was a disease that could lead to progressive fibrosis, cirrhosis, and liver-related death. At this point, we now knew the clinical severity, but we did not know, we just could not. We tried every serologic, enzymatic, radioimmunologic test and very early molecular methods to get a specific test, and it just did not work out. So it was at that time that I wrote this poem of frustration that I called "I Can't See the Forest for the HbAgs." It went, "I think that I shall never see this virus called non-A, non-B, a virus I cannot deliver, and yet I know it's in the liver. A virus that we often blame, but which exists alone by name. No antigen or DNA, no little test to mark its way. A virus which in our confusion has forced us into mass collusion to make exist just by exclusion. But is it real or an illusion? Oh, great liver in the sky, show us where and tell us why. Send us thoughts that will inspire us. Let us see this elusive virus. If we don't publish soon, they're going to fire us. Well, this poem, which I presented at a national meeting, I think had a lot of influence on Michael Houghton and the Chiron Corporation, because soon thereafter they cloned the non-A, non-B agent. Now you're going to hear from Michael Houghton, so I'm not going to go through this in any detail, but I just want to point out what a great study this was. Chiron and Michael and his group extracted RNA from high titer plasma obtained from a chimpanzee and also from chronic carriers. They took this RNA, reverse transcribed it into cDNA, cut this up with restriction enzymes, and importantly, injected this material into a phage GT11 expression vector. This was a bacteriophage that would not only carry genomic information, but would express the protein coded for by that gene. They put the phage into E. coli, let it grow, then lyse the E. coli, absorb the lysates on filter paper, and then made another important step, observation. Michael assumed that there would be antibody, even though nobody had ever found antibody to non-A, non-B up to that time. But he said there's never been a virus that doesn't induce an antibody. The story goes that Keelam Chu and the group looked at 6 million clones before they found a single clone that was reactive by this methodology, but they were then able to subclone it, express a protein, a small protein, to walk the genome, and eventually have enough material, enough antigen to develop an antibody assay. This was a remarkable step at this point in the evolution of molecular biology. I always thought it should get a Nobel Prize, and I'm happy that it did. Here's Michael Houghton on the right. At the time he received the Lasker Award, and Dan Bradley on the left, who worked closely with the Chiron Corporation. Dan was at CDC. Michael will tell you more about him. When Chiron felt they had a non-A, non-B assay, which they now call hepatitis C, they asked for the non-A, non-B panel that I had developed. This was a small panel, but it had baffled 19 other laboratories who claimed to have a non-A, non-B assay. In the panel, all the samples were proven infectious in the chimpanzee model, and within in the panel is duplicates in random positions. There were also negative controls who were patients who had donated at least 10 times and never been implicated in a case of post-transfusion hepatitis. The Chiron Corporation broke this code perfectly. They detected all 3 chronic non-A, non-B cases in 6 duplicate samples. They detected 2 implicated blood donors in 4 duplicate samples in random positions. They did not detect 2 patients with acute non-A, non-B because they were measuring antibody, not virus, but later these patients seroconverted for antibody. And importantly, they did not detect the agent in any of 7 negative controls in 14 different positions. This was the key for specificity. Thus, they had discovered or cloned hepatitis C virus. We then went to our own serum samples, looked at 15 non-A, non-B cases, found that in 100%, the patients were negative for HCV pre-transfusion and became positive post-transfusion. We then looked at 25 non-A, non-B cases, looked to the donors to these cases, found a positive donor in 80%, by a first generation assay and 88% with a second generation assay. And thus we could predict that if we introduce this test into blood donor screening, we might prevent near 90% of post-transfusion hepatitis. Indeed, that's what happened. We started the lecture with this early point with a very high rate. We did other interventions over time that helped decrease the incidence, particularly lowered the amount of blood that was transfused. But by 1990, at the time Chiron made the discovery, the rates had come down to 4.1%. But with the first generation assay, it dropped to 1.1%. And by 1995 to 97, we were down to 0 incidence of post-transfusion hepatitis. We can't say it's absolutely 0 because this is a relatively small study, but the risk now is mathematically calculated to be 1 in 2 million. So it's come from 1 in 3 patients getting hepatitis to 1 in 2 million by 1977. And in fact, we have not seen another case in another 1,000 patients of hepatitis C or B post-transfusion. It's estimated that in the US, we may have transmitted, that blood transfusion may have transmitted 4.8 million HCV infections in the 2 decades from 1970 to 1990. And conversely, that with the introduction of the test in 1990, that we probably prevented 2.4 million transmissions in the next 2 decades. This is the same slide now with just this downward arrow, and I show this because this is not just a story of post-transfusion hepatitis. This is a story of my life. And you can see that both post-transfusion hepatitis and my life are in very rapid decline. But I'm happy to say that hepatitis seems to have disappeared, and I'm still here talking about it. So that is the end of the beginning, the eradication of post-transfusion hepatitis, the development of this very sensitive assays. But there's a big story beyond that. As you know, there's about 70 million people in the world who are chronically infected with hepatitis C, 350 million chronically infected with hepatitis B. But with hepatitis C, the important point is 75 to 85% of people who have HCV infection go on to develop chronic persistent infection. And we were interested in the reasons for this. And working with Patricia Farsi here at NIH, Patricia Farsi took the inoculum from Mr. H and cloned studied 105 clones. She sequenced these clones. 57% were identical. This was the dominant strain, but another 19 clones were present at the same time in this single blood sample. So that even if one developed a good antibody to the dominant clone, any one of these others could have emerged to take over and keep the infection going. To show you how this works, we looked at patients during the early phase of infection. We looked at 4 points prior to the peak of the ALT, did 10 clones at each point, and at week 3, in these 10 clones, you can see there were 3 variants, arbitrarily labeled A, B, and C, with A and B being kind of co-dominant. But by week 8, now D was the dominant strain, but there was also E, F, and G. By week 13, you could see A was still there, but no longer dominant. Now there was H, I, J, K, L, N, O, P. And by week 16, all the way down the alphabet to U. So you can see this continual emergence of variants in what's called the HCV quasi-species. So people don't have a single HCV virus, they have a whole host of HCV viruses in their blood at any given time. Another reason for the persistence in the study done by Wertheimer, this is not from my own group, but it's very instructive. He showed that patients who had chronic hepatitis C had no CD4 or CD8 responses as shown by an ELISpot assay. They were no different than normal patients who had never seen hepatitis C. On the other hand, when they recovered from their infection, as 25% of patients do, they had good CD4 responses and good CD8 responses. So the T cells were not killed in this infection, but they were blunted by the high viral load. So the quasi-species and this poor T cell response is what keeps the infection persistent. I don't have time to go into many other aspects of this, but I just wanted to talk about one more thing, which is liver cell cancer. This was a study we did with Japanese investigators, Dr. Yasuhiro Tanaka and Dr. Mizukami, where we compared genotype 1b in Japan, shown here in the red, with genotype 1a in the US. Now, the molecular clock that Dr. Mizukami and Tanaka performed is a mathematical modeling of varying sequences in a patient over time. And that allows one to go back and tell when a particular genotype emerged from some common ancestor, and then when it spread rapidly within a population. Well, you can see in Japan, genotype 1b diverged from a common ancestor around 1880, and then was relatively stable until the 1930s and 40s when it rose rapidly in Japan. But what was going on in Japan at that time was that they were at war, first with China and then with the rest of the world. And we learn now in retrospect that Japanese soldiers were shooting up prior to battle using shared needles with amphetamines at that time, but they were sharing needles, and this was the cause of the high rates of hepatitis C in Japan. But those are now leveling off as this population is reaching old age and dying off. In contrast, in the US, genotype 1A emerged in 1910, was very stable over time, but then rose in the 1960s and 1970s and 80s when we had our own drug epidemic in the US with shared needle exposure. The importance of this is that there's a 30 to 40 year difference between these two curves. And that's important because the rates of hepatocellular carcinoma in Japan are 8 to 10-fold higher than they are in the US. And the Japanese always advise us that their rates are so much higher because they've had the virus for a longer time. And that once we had the virus, as long as they have, our rates of hepatocellular carcinoma will also rise. This has proven to be the case. In this study by El Sarag at Baylor shows that there is been a threefold rise in the rate of hepatocellular carcinoma from the late 1970s through the late 1990s, and that this is all due to hepatitis C, not to B or other causes. It was assumed in 2009 that if there were no changes in the standard of care, that the total number of patients with advanced liver disease by 2029 would be fourfold higher than they were in 2009. And this was what's going to happen, was going to happen, but there has been a dramatic change in the standard of care. We now have these wonderful drugs that have evolved, these direct-acting antivirals, which now give a 95 to 100% cure rate of hepatitis C. It's just been a real miracle how easy and how effective treatments are. So with these great treatments now, question arises, well, let me just say first that you can see hepatitis C at this point in 2021. You can see that the glass half full or the glass half empty. If you see it as a glass half full, you can say with cure rates now approaching 100%, from this time forward, once HCV infection is identified in a patient, no patient should develop cirrhosis or die from hepatitis C sequelae. But if you see the glass is half empty, then you know that only about 50% of infections have been identified. That throughout the world, many, many people are still chronic carriers of hepatitis C and do not know they're carrying the virus. And this is particularly true in the developing world. Secondly, access to treatment is not uniform throughout the world. And even among known carriers, early on, only a minority got treated with these great drugs because the drugs were so expensive. Fortunately, the proportion being treated now in the developed world is increasing very well as drug prices come down. But still, there are many areas of the world that do not have access to treatment. The high cost of drugs has definitely been an impediment to treatment, preventing the cure of millions. So cure of hepatitis C is no longer constrained by science, but really it's a matter of dollars and the will to make it happen. So we could say, can the HBV and HCV infections be eradicated? Well, for HBV, we have a great vaccine. It's greater than 95% effective in preventing hepatitis B infection. We have nucleotide and nucleoside inhibitors, which can drop the viral loads down to undetectable and prevent transmission. But for hepatitis C, we do not have a vaccine as yet. Michael Houghton will talk about this, but we do have these great direct-acting antivirals. So if we want to go to global eradication, we just need a little miracle. But this miracle has some measurable components. First of all, for B, we could do this universal vaccination, prevent infections in the newborn. This is moving along throughout the world, and HBV could be eradicated. But for HCV, it's going to depend on detection followed by treatment. So we have to detect almost all HCV carriers through massive global screening. And then we have to do this with a rapid test so that we can deliver the direct-acting antivirals immediately to the patient and need to penetrate probably at least 90% of each population. So eradication is theoretically possible if we had the political, the corporate, the philanthropic, and the moral will to make it happen. But it's not going to happen on my watch. It's going to be up to you young people who are listening to this lecture to make this happen country by country, to get this testing done, to get the treatments administered. Egypt has already done this. Egypt had very high rates of hepatitis C, and they have been able to almost test everybody in the country and treat everybody who needed to be treated. This has to be done, and I'd like to see Portugal be next on the list. But it's not going to happen with me because I have a new philosophy of age, and which I have here on the abscissa, and I plotted that out with give a damn. And you'll see I'm way out here on the curve. I'm fading away. I do still give a damn, but I'm not going to be that involved. So I hope you young people will take on this challenge. So I just want to end with another poem that I wrote called An Ode to HCV. Your days are numbered, my fine feathered friend, with drugs now attacking your enzymes and your 3' end. Drug after drug is on your case, snuggling up to your polymerase. See how well you do without your protease. Your non-structural proteins are now big targets. They're selling your pieces on the open market. More than half your victims are already cured, and 90% responses the future assures. You were quite proud of your force of destruction. Killing hepatocytes was your seduction, disabling lymphocytes as if by abduction, stopping coag factors en route to production, shrinking the liver by fibrotic reduction. HCV, you're on your way out. A few more years and you have lost your last shout. You've had your last shout. At meetings you will no longer talk about. You'll be a virulent, have lost all your clout. Of your ultimate demise, there is now little doubt. In many ways, I'll miss you. You've been a good foe. We've waged a long battle, gone to and fro, and along the way, you've improved my cash flow. I respect your persistence, your state incognito. You've aged like a fine Bordeaux, but now you must rest and bask in what was your glow. We've got you, you bugger, and will soon wear you low. Singing your swan song is now apropos. So thank you very much, Porto, and I will see you again in the discussion section.
It's my pleasure now to introduce Professor Michael Houghton, and he was in the history of the discovery of hepatitis C virus, as you will see. We'll give a talk immediately after this short introduction. He was born in United Kingdom and completed the PhD in biochemistry at King's College, University of London in 1977. He was involved in the study of human interferon gene regulation before moving to Chiron Corporation. You have listened already about this Chiron Corporation. That was, in a way, the environment in which the studies for the identification of hepatitis C virus were performed. So he published, was co-author of many seminal studies in 1989 and 1990. The virus was identified in 1989 that identified hepatitis C antibodies in blood, particularly among patients at higher risk of contracting the development of a blood screening test in 1990. So the identification of these antibodies was crucial for the development of serologic tests. His work led in 1992 to the development of more sensitive tests, which has eliminated hepatitis C contamination of donated blood supplies in Canada and around the world. His group also identified key enzymes in the viral replication cycle that have been the target of successful drug development by international pharma companies. Here I have to say that were not Harvey Alter and Michael Houghton those who received the Nobel Prize in 2020. There was a third person, and this third person is not attending this session today, is Charles Rice, and he played a pivotal role in the identification of the structure of hepatitis C virus. So the three of them, and before giving the word to Professor Michael Houghton, we want to thank so much for the privilege of these young students being able to listen today to their talks. Thank you so much.
Hello. Well, the hepatitis C virus field has been going around 30 years now. And so I think the organizers asked me to project what's going to happen over the next 30 years. So thank you for that invitation. I think the WHO recently issued objectives for curbing and containing viral hepatitis. The goal of their objectives is to eliminate viral hepatitis as a major public health threat by 2030. Hepatitis B and hepatitis C are responsible for an estimated 1.4 million deaths every year around the world. Hepatitis C contributes an estimated 400,000 fatalities every year. The specific goals of the WHO to contain and eliminate viral hepatitis as a public health issue are as follows: a 90% reduction of new cases of chronic hepatitis C, a 65% reduction in mortality, and the availability of more sterile needles and syringes provided to people who inject drugs. There's still not enough sterile equipment available to people who inject drugs, which of course is the most major source of infection. WHO want all blood supplies around the world screened in a quality assured manner by 2030. It is surprising that after 30 years of having tests available, even today, such screening is not carried out in all countries. We need to fix that for sure. WHO also would like to see 90% of injections inside healthcare facilities and outside administered with safety engineered devices all around the world, very importantly to prevent needle stick accidents, which could transmit hepatitis C, and for 90% of people with hepatitis C virus persistence to learn of their status. If they can know that they're carriers, then they can be treated and cured. And last but not least, WHO want 80% of all people with chronic hepatitis C to be treated with the antivirals now available, which are extremely effective, well tolerated, and cure the vast majority of patients within 2 to 3 months. In Canada, our Canadian network on hepatitis C has issued a blueprint to indicate how Canada can achieve these goals by 2030. All countries need to have their own national plans for achieving the WHO goals. Harvey Alter recently let me know about the fantastic success that has happened in Egypt in testing their populations for hepatitis C and then treating them with the new modern antivirals. An excellent review has been published recently in Global Health by Dr. Hassanin and colleagues. And what this review describes is an elimination campaign between 2014 and 2020 that has successfully screened more than 50 million Egyptians and treated 4 million carriers that were identified with the very potent new antivirals. This is a tremendous success story. It's still ongoing, so that all citizens in Egypt will eventually be tested and treated. But this has been a tremendous model for all countries around the world in testing and treating and eliminating hepatitis C disease. The Egyptian model was greatly facilitated, I think, by the cost of antivirals being very low. The actual cost, as you can see, was around $84 per patient or less. Current drug prices in the West are much higher, in the region of 20,000. So it will be important in Western countries and in other countries around the world to reproduce Egypt's model to have lower costs for these very powerful and potent antivirals. While identification of hepatitis C carriers followed by DAA treatment is a crucial policy worldwide and clearly has been demonstrated to work very effectively in Egypt, if we can produce an HCV vaccine, this would greatly facilitate and also accelerate the attainment of the WHO goals. We have learned that both neutralizing antibodies and cellular immune responses to the virus correlate with immunity. There have been several reviews. I recently reviewed this in a Cold Spring Harbor Perspectives article. And I think with the technologies that are available through vaccine methodologies, including the new ones developed so well and so quickly for COVID vaccines in the form of RNA and adeno-based technologies, we have the ability to recapitulate these protective immune responses via vaccination. So I think a hepatitis C vaccine is very feasible in a matter of a few years. In terms of correlates of immunity, cellular correlates have been defined extremely well by the laboratory of Chris Walker in Ohio State. Using the chimpanzee model, Dr. Krakui depleted the entire CD4 T cell compartment by antibody treatment, and then challenging two chimpanzees after depletion led to persistent viremia, work that was published back in 2003. And so while these animals in undepleted form were able to spontaneously resolve an experimental infection, once the CD4s were removed, following rechallenge, they became persistent carriers. This is direct evidence that T helper responses to the virus are required for immunity. And then also in Chris's lab, Nagla Shukri, now chair of CanHepC, did a similar experiment in chimpanzees where she depleted not the CD4 compartment, but the CD8 T cell compartment with antibody treatment. And once again, she enrolled two animals that had been able spontaneously to control infection. Then she depleted CD8 T cells, then challenged them again. And what she observed was a longer duration of viremia than in the first challenge. And these animals did not resolve the second infection until the antibody declined and the CD8 T cells started to re-emerge. So this is once again direct evidence that CD8 T cell responses to hepatitis C are required for immunity. When I mentioned antibody was to the CD8 T cell marker to remove all CD8 cells. You can see in this animal, once multifunctional CD8 T cells returned to the liver, then they controlled the viremia and ultimately its resolution. In this animal, there were multifunctional T cells, CD8 T cells, not as broad as this animal, but nonetheless, when they reappeared, that was when control of viremia took place and eventual eradication. The situation with antibodies has been somewhat controversial, I think partly because it took longer to develop neutralizing antibody assays for hepatitis C. But I think there has been for quite some time strong indications that neutralizing antibodies are playing a protective role against hepatitis C infection. This is a slide from a paper published from Thomas Beaumart's lab back in 2007. Dr. Pesca looked at the presence of neutralizing antibodies early on in acute infection, and what he saw was a much higher level in individuals that went on to spontaneously eradicate the virus versus individuals that went on to develop chronic persistent infection. And this difference in neutralizing antibody titer was highly significant, shown by this p-value. Also, Dr. Jean-Pierre Michael Pawlowski showed similar findings in some of his cohorts of patients where resolution of acute infection correlated with the induction of neutralizing antibodies. A direct demonstration of the potency of neutralizing antibodies came from use of the Alberta mouse model, as I like to call it. My colleagues at the University of Alberta, Dr. Tyrell and Dr. Kniedemann, and individuals in their labs, developed a SCID UPA mouse model, which allowed the colonization of mouse liver with human hepatocytes. And then those colonies of human liver tissue could be infected with hepatitis C as well as hepatitis B, incidentally. This is a great model. Dr. Meulemann's group in 2011 used this model to show that antibodies purified from Harvey Alter's famous patient H, this is a chronically infected patient, that those antibodies, when they're passively transferred into this mouse model prior to challenge with virus, was protective. And you can see here when the mice were passively immunized with the IG from patient H, then challenged with homologous virus, the 1A homologous virus. The red symbols are the passively immunized animals, and the blue symbols represent control mice. And you can see that for 3 weeks thereafter, the passively immunized animals were completely protected. When Dr. Meulemann did similar studies, except challenged the mice with heterologous 4A and heterologous 6A, which are highly divergent strains of HCV, the protection wasn't quite as impressive as against homologous. However, if you look at the horizontal lines for both the experimental and the control groups, you can see that there was a major impact on viral RNA levels between the passively immunized animals and the control animals shown in blue. And 2 weeks, 3 weeks after challenge, even with these very high heterologous isolates, 1 or 2 of the mice were still PCR negative. It's important in looking at this data to understand that the half-life of the human IG is very short, only around a week. And so it's rapidly declining. And I think that needs to be taken into account. And I think further enables the conclusion that this experiment and others like this that have already been published are direct evidence for the potency of neutralizing antibody in HCV immunity. Dr. Moran in 2012 used the chimpanzee model. This time he used a monoclonal neutralizing antibody called HCV1. And if he passively immunized a chimpanzee with high enough levels, 250 mg per kg, he was able to completely prevent infection following experimental challenge. The controls are shown in red and, in green, and the red is when he used a 5 times lower level of antibody. The animal got infected, but if you use a high level of antibody, he was able to completely prevent infection. So once again, another direct demonstration of the role of antibody in protection. At this meeting, we have heard about many approaches to developing a vaccine, and I think we have many ways to do this, and I think it's very feasible. As I mentioned, I and colleagues have focused on the use of recombinant envelope glycoproteins E1 and E2 as a vaccine candidate. These are produced in CHO, Chinese hamster ovary cell lines. When you co-express E1 and E2, they form a heterodimer that is very tightly locked into the lumen of the ER, very tightly anchored. The heterodimer does not get out into the media. And over the years, when I was working in the US still, we were able to show that if we immunize chimpanzees with adjuvanted E1E2, highly purified E1E2, we could show that we could protect these animals against chronic persistent infection. And to my knowledge, it's the only HCV vaccine candidate that has been shown to be protective in animal models. And just quickly to summarize the data, which probably many of you have already seen for many years, you can see that when we challenge with homologous virus, only 17% of animals became persistently infected versus 70 in control unimmunized animals. This was statistically significant. When we challenged 19 animals that were vaccinated with heterologous 1A virus, only 16% became persistent carriers versus 57% of control unimmunized animals. Once again, statistically significant. And then if you put all the data together, it clearly shows that we were able to substantially reduce the incidence of persistent viremia. Since clinical disease is associated with virus persistence, this indicates that this is a very strong vaccine candidate. Another concern about hepatitis C vaccination is because the virus is so heterogeneous, more heterogeneous than HIV, would it be possible to deal with that heterogeneity by vaccination? But in work that we did with the NIH 10 years ago in Bob Purcell's lab, Dr. Munier took our vaccinated chimpanzees that were protected and asked the question, does that antisera neutralize the various genotypes of HCV observed around the world? And he published that he could see such breadth of neutralization. We gave him sets of 4 animals, and you can see that he was able to show significant neutralization of infection of genotypes 1, 4, 5, and 6. Genotypes 2 and 3 were not as well neutralized, but nonetheless, there was still significant neutralizing activity observed against those genotypes. My colleague, John Law, who I've been working with for more than a decade now, an excellent virologist, he addressed the same issue in humans that we had vaccinated with E1E2 along with the MF59 adjuvant. This was a phase 1 trial run by Chiron when I was still there in the US and funded by the NIH. The vaccine was shown to be well tolerated and immunogenic in terms of overall humoral and cellular responses. And John, in 2013, published his data where, once again, selecting 3 individuals from that clinical trial who were good responders to the vaccine, he was able to demonstrate that they did indeed elicit a broad cross-neutralizing antibody response against all the major genotypes of the virus that occurs around the world. We're very grateful to Jens Book and colleagues for providing these chimeric viruses that we were able to grow in cell culture. And you can see, for example, individual number 5 had a very broad cross-neutralizing profile. This is with only using MF59 adjuvant. It's not a particularly strong adjuvant. Now we can use stronger adjuvants and intend to do that in phase 1 clinical trials next year. But importantly, this was a seminal finding, I believe, by showing for the first time that a vaccine derived from a single strain in man can elicit broad cross-neutralizing antibodies. Excuse me. We understand why that is so, and that is because we've identified multiple cross-neutralizing epitopes within E1E2. Grateful for the collaborations we had with Stephen Fong at Stanford, Manson Law at the Scripps, Dr. Diamond, and other collaborators who donated their cross-neutralizing monoclonal antibodies to us to interrogate E1, E2. This is a slide showing the atomic structure of E2 that was worked out back in 2013 and 14 by Joe Marco Trigiano's lab as well as Manson Law's lab. We don't have the structure of E1 or E1, E2 yet. That will be very helpful if we could identify that soon. But this diagram shows that the various cross-neutralizing monoclonal antibodies identified by various groups bind to different regions of E1 and E2. And these can be linear epitopes like the ones downstream from the N-terminus of E2, or they can be discontinuous epitopes within E2, and also very important epitopes that are discontinuous and require the presence of E1 and E2. These monoclonals that are very high affinity and broadly cross-neutralizing do not bind to E2 alone, do not bind to E1 alone. They only bind to the E1, E2 heterodimer. It's one of the reasons I've been very keen to develop the E1, E2 heterodimer as a vaccine antigen. The other reason, of course, is that we've got clear evidence for protection in the reliable chimpanzee model. Our student, Jason Wong, was able to use these monoclonal antibodies generously donated by collaborators around the world and show that in the phase 1 clinical trial in which volunteers were immunized with E1, E2 plus MF59, he was able to see clear evidence for those volunteers eliciting antibodies that could compete with these cross-neutralizing monoclonal antibodies. In this particular slide, Jason used 4 of the antibodies, including 4A and 5A, that require both E1 and E2 for binding and neutralization. Before vaccination, there was no evidence for inhibition of binding of these monoclonals. After vaccination, in 5 individuals that Jason interrogated, he could see clear evidence for inhibition of binding of these 4 broadly cross-neutralizing monoclonal antibodies. So although the virus is very variable and there's sequence divergence of more than 30% at the nucleotide level, nonetheless, within the 2 envelope glycoproteins, there are multiple highly conserved cross-neutralizing epitopes. And you can recapitulate those antibodies by vaccination. Well, so we're developing an E1, E2 adjuvanted vaccine. We want to broaden and also focus the cellular immune response. The Illustrious vaccine was shown not to be efficacious in preventing persistent infection in people who inject drugs. Really nice work by Dr. Cox and Dr. Page and colleagues in a heroic study were able to actually test the vaccine very efficiently and very well. But unfortunately, the T-cell vaccine that was elicited against a very large polyprotein encoded by HCV in the absence of neutralizing antibodies was not shown to be efficacious. However, as I mentioned earlier, we do know that CD4 and CD8 T-cell responses are required for immunity. So what we are doing is focusing the immune response, the cellular immune response, on highly conserved T-cell epitopes. My colleague Amir Landi did a bioinformatics exercise and carefully came up with a cocktail of 4 synthetic peptides that we call TPs, that contain multiple conserved CD4 and CD8 epitopes. These are conserved almost amongst the entire genus of Hep C virus. And furthermore, he came up with these 4 peptides to ensure that there would be a CD8 presentation restricted by MHC1 in the vast majority of individuals that we will vaccinate. And that's why you see such a heterogeneity of MHC class 1 restriction to ensure that we get good CD8 responses in more than 90% of individuals. So we think it's good to broaden the T-cell response, but also to focus it on very highly conserved epitopes. Chris Walker has very nicely shown the virus can evade T-cell responses, CD8 positive T-cell responses, but by providing T-cells and eliciting T-cells to highly focused epitopes, that is going to be very hard for the virus to evade those. Of course, we need a good adjuvant that can prime good antibodies as well as cellular immune responses using these subunit proteins and peptides. And I'm just going to show you some work that we've done recently on the receptor binding domain of the COVID-19 virus. We made the receptor binding domain in CHO cells, my colleagues Mike Logan and John Law. And then we immunized mice with various adjuvants. We saw good antibody levels, neutralizing antibodies. And my colleague Amir Landi identified and monitored and quantitated the cellular immune response. And I don't want to dwell too long on this figure, but what it shows is that, for example, here with this particular adjuvant here, we were able to see very strong polyfunctional T helper responses to the receptor binding domain of COVID. These are T cells specific to RBD that are secreting both alpha, TNF alpha, and interferon gamma. We also showed they are secreting IL-2. Not only that, but the right adjuvant can indeed prime CD8 positive T cell responses to subunit proteins. And you can see with this particular adjuvant, Amir recorded almost 1% of the CD8 T cell population being specific for the receptor binding domain of COVID. So choosing the right adjuvant is extremely important, and we intend to do that in phase 1 trials next year. So unfortunately, COVID, partly because our research has actually been transferred to COVID away from Hep C to some extent, and partly because of the lockdowns we've endured in Canada, and also supply chain problems, which are real and still are. Our timelines for going into phase 1 trials with our Hep C vaccine have been delayed. We anticipate going ahead with phase 1 trials next year now, using different adjuvants. We want to see, it's all very well testing in mice or in rhesus macaques. You don't really know how humans are going to respond until you test it in humans. So we're going to test 2 different adjuvants in phase 1 trials next year. If it's safe, which we anticipate based on previous clinical trial experience that I've had with Hcv vaccines, and if we can get broadly cross-neutralizing antibodies and strong CD4 and CD8 T cell responses, we will then go into efficacy testing in humans. Now, efficacy testing can be done, as in the case of the Arcturus vaccine, in people who inject drugs. Drs. Cox and Dr. Page did a really good job running those trials. So probably that will be the way we go. However, there's been a lot of discussion recently about the prospect of vaccinating human volunteers, challenging them with the virus, and if they become persistently infected, treat them with the antivirals. So vaccine challenges are performed quite widely on a number of different pathogens, for example, malaria, dengue virus, and others. Now that we have these really potent antivirals against Hep C, it is possible to think about getting ethical approval for such vaccine challenges with Hep C. So if we can do a vaccine challenge, it will speed up the development of the vaccine. Now, we have regulatory consultants, and what they tell us is that if we can conduct a trial in people who inject drugs, show that it's safe and efficacious in those folks, we will be able to roll out the vaccine to that high-risk group straight away without having to to go through phase 3. Now, we obviously would have to get regulatory approval to do that, but because of the high risk that people who inject drugs are under, in a phased process, we should be able to start rolling this vaccine out to that high-risk population around 2026, 2027. I think in the literature, there is a general agreement that a HCV vaccine not only would be extremely useful and powerful at curbing the hepatitis C pandemic, which is exactly what it is, but also it would be highly cost effective. In Canada, if you figure out the number of carriers that will be generated in people who inject drugs over the coming decade, and if you treat them with antivirals, which is a good strategy for sure, but the cost will be very large given the current prices in the West. Even if you account for discounts in the future, we are looking at costs, antiviral costs of $1 billion approximately, versus just 20 million if you vaccinate those high-risk people who inject drugs. So medically, it will be extremely powerful as well as very cost-effective. So I think that's the prospect that we're looking at in the mid-2020s. Hopefully, we can roll out vaccines to the people who inject drugs around the world and thereby drastically affect and reduce the incidence of infection and disease. I think to get it to males who have sex with other males, healthcare workers, babies born to positive mothers, we will have to do phase 3 trials. So this will take another 2 to 3 years. But I think even then, by 2030, we can expect a broadly applicable vaccine to be licensed. And there may well be different vaccines approved in different parts of the world. And of course, we need to roll out the vaccine to low and middle income countries where HCV prevalence and incidence is much higher than the West. That's going to be crucial to achieve the WHO goals. After 2030, we will need to support the global delivery of the antivirals and the vaccines. You know, I think we all know that antiviral delivery for Hep B and HIV has been effective, but we would like it to be faster with Hep C. The Hep B vaccine was certainly taken up quickly as a universal vaccine. We would like a rapid deployment of any Hep C vaccine that is developed in this decade. And of course, once we achieve that, then Hep C really will be eliminated as a public health emergency. And then we can apply what we've learned on hepatitis C to many other important viruses that afflict mankind. I would like to acknowledge many excellent scientists that I've worked with at the University of Alberta on the vaccine for the last 10 years. John Law, Mike Logan, Amir Landy have been really outstanding contributors to the program, as has our lab manager, Darren Hockman, who seems to do everything at all times. We've had excellent research associates contributing to the program, excellent students, many of whom are now members of our GMP manufacturing team, and they're really enjoying doing that work. We have other specialists. I brought in Kevin and Mark from Chiron and Novartis, who are GMP bioreactor specialists and cell line specialists. We've had really brilliant technical contributions from Chow and Drew, and we have a very excellent QA and QC team to monitor the quality of our vaccine going into humans soon. Last of all, I'd like to acknowledge and thank my colleague and friend, Lorne Tyrell, not only for his collaboration and friendship, but also he has been a co-PI on our CREO grant to the Alberta government, and also a co-PI on our Government of Alberta grant to our Applied Institute. Thank you very much again for being able to talk to you, and wish you all well, and look forward to eliminating hepatitis C with you by 2030. Bye-bye. Take care.
we began with micro-elimination programs, and now we're moving to the elimination national program. Anyway, there are some burning questions from our audience, especially to you, Professor Harvey Alter. Nice to see you again. And there is one particular question that is striking me. I think this is the $1 million question, which is, what are the main factors that really are important for the development of hepatocellular carcinoma? Is it the virus itself? Are there host factors? Is the inflammation process? Why are there so much HCCs in hepatitis C?
Yeah, that's a difficult question. Almost all the hepatitis carcinoma evolves in people who already have cirrhosis. It's not uniform, but I think it's somehow in the process of inflammation, fibrosis formation, and regeneration, attempts at regeneration, that mutations occur. But what are the critical pieces that generate those mutations or that arouse a oncogenic gene? I really do not know. But we do know that when you are treated, when the inflammatory process stops, the risk of developing hepatic carcinoma goes dramatically down.
Right.
And probably the people who go on to have a carcinoma after being treated to cure probably already had that process started already. I don't know whether there's any direct viral effect. I don't think so, truthfully. I think it's similar to what happens in hemochromatosis and alcoholic liver disease and any disease that goes on to cirrhosis. But that's a key question, really, and it's a good question.
Great. Professor Fatima?
Thank you so much for this presentation. I think this was real enlightening for the students at this phase of their training. And I have a very short comment that is related with the previous question. In my mind, in comparison with hepatitis B virus, the major difference is the quality of the DNA for hepatitis B and RNA for hepatitis C. So there is no integration in the genome. Do you agree this can direct different settings of developing the hepatocellular carcinoma?
Yes, I do. There is definitely no integration in hepatitis C. In B, you have the same process to cirrhosis, but in addition, you have the integrations that may set off an oncogenic gene. But true, in C, that's not the case.
Yeah, absolutely.
One thing that is striking our students here is that you both mentioned the Egypt's way of doing it, the strategy they had, which has been praised all over the place. But the question is, why or what made their action so effective? And what can the other countries do, either in the, let's say, high-profile countries or the low-profile countries? What can we replicate from the Egypt's example?
Well, I think it starts from the top. Egypt felt some responsibility for having caused the hepatitis C through the vaccines for schistosomiasis, which infected a huge number of people. The decision was made that we have the highest rates in the world because of that. and what can we do? And the government got behind it and said, well, we have great drugs, we have good tests. All we have to do is test people and treat those who are positive. That's the strategy short of a vaccine. And it takes an enormous effort, enormous national effort to make that happen. But if you decide that's what you want to do, it is feasible to do it. And you can do it, we could do the world country by country, or you could do a country region by region. But we know it works. It's a very unique situation where we have such highly effective and safe drugs.
Yep.
Whose cost has come down, but they're not inexpensive. But that's, I'm hopeful that Michael or somebody will come up with a vaccine that will speed the process. But in the interim, Egypt has shown it can be done. Brazil is trying to do that now also. So you have to have a national will to make it happen. You have to have the financial resources to provide the tests and the drugs. But I think one key is to have a rapid test so that when you have the person with you, and they test positive, you can immediately give them the first month or 2 months supply of the drug. So you don't have that loss of between testing and treatment.
You're absolutely right, and that's one of the things we are doing here right now. But again, the main and the critical issue is the political will. Unless the politicians are not aware of this disease, and in Egypt they were, because they found to be, let's say, responsible for the epidemic out there. So there was a sensitivity about that. They were sensible for that. So the crucial thing is our politicians to be aware that they should spend some money, not too much money, because we don't need to spend that money. For example, for our control of HIV infection here in our country, we use around 200 million euros per year, every year. And we know now, and we contributed to that with 200 million euros just for 5 years, meaning 40 millions per year, we could eliminate hepatitis C. So the problem is not financial, the problem is political will, and that is really the question. Now, I have one last question that they made us, and in fact it will be the last question. And there was one of our students asked, perhaps he's not aware of Pablo Neruda's Nobel Prize, and Pablo Neruda wrote a marvelous poem about the liver. But anyway, he was or she was asking that viral infections and chronic inflammation generally are not common topics for poetry. Where does the need to express your work through this medium come from?
I don't know. I started writing poems when people retired from work, and I would write a poem in their name and their honor. And then I did some for meetings, and they always seemed to be popular.
Exactly.
So I've just continued to do that. But this one about the Nobel Prize, I think really expressed my true feelings. I did not expect to win a Nobel Prize. I would not have won the prize had Michael not cloned the virus and developed the tests for it, and Charlie Rice and others developed the treatment. So it was all three pieces together that made this nice story. But I guess I just I found that rhyming on whatever topic I'm talking about gets a good reception.
Exactly, that's absolutely true. And again, I was there when you had some of these poems quoted in the ASLD annual meetings, etc., etc. That was a great experience. Well, thank you very much. We were pretty much honored for the privilege that having you and Michael Houghton presence in virtual presence and teaching us with your conferences how were your experience throughout these years in this, well, could be a deadly combat. It is a deadly combat for the virus, hopefully. So, but again, that really changed the history in mankind. And so I think we are all very fortunate to have the chance of listening all these to magnificent conferences from you. So thank you very much again. And also thanking Professor Fatima for having this co-chairing with us. And again, showing our students that there is a bright, large, and big world of science out there that merge with the clinical aspects of what we do and really come up to change the world as you did. Thank you very much.
Thank you.
Thank you. Thank you.
This is the end of the Nobel Prize session. We would like to thank Professor Harvey Alter and Professor Michael Houghton for their magnificent lectures, which were followed by a wonderful coordination of the discussion by Professor Fátima Carneiro and Professor Guilherme Macedo.